Welcome to Hidden's Physiology channel. Today we're going to be talking about muscles. Not my muscles in specific, you gotta stop staring. We're going to talk about the intracellular muscles, stuff that's going on the inside of your body. So we got to break this down into pieces. First and foremost, I want to talk about the big picture. Now you may be saying, what in the skeletal heck is going on here? That's actually not a skeleton. This is a muscle on the inside. So let's do some labeling, because who doesn't love some good labels? That's just me grabbing a marker. You can see that if you want. So this blue stuff here, this is going to be our myosin, is what we're going to call it. And then this red stuff here is going to be our actin. So what is essentially happening here is we have our myosin, which we call our thick filament, and then we have our actin, which is our thin filament. And they're going to adopt something called the sliding filament model. So although I can't illustrate this because this is a whiteboard and I can't move them back and forth, you can imagine that these actin are going to squish in and out like that. Now when I say actin are squishing, what's actually happening is this myosin is pulling on it, pulling in both directions to cause the actin to contract. Now believe it or not, this makes up the vast majority of the inside of pretty much every cell, especially muscle cells, so that way they can contract and do stuff. It's just sort of it's all inside the cell. And this diagram here does have some labels as well, and I'll define these, and I'll have to go through and define these. So this line right here, we call it the M line because it's in the middle. And then these zigzag lines over here are actin, we call these the Z line. And I call it the Z because it looks like a Z. I think that's probably why they call it that. And then over in this region that only includes the actin, we have the I line. Let's see, is it H or I? I band. And then over here, again, there's also the I band. And in the middle, we have the H band. So the I band only includes the actin filaments, and the H band only includes the myosin. So both of these grow and shrink with contractions. And then right here, that encompasses the entire myosin molecule. We call this the A zone. Now this one does not grow or shrink, it's entirely just encompassing the entire molecule. If we were to see this on like a TEM, which is a topographic electron microscope, two-dimensional, we would see this has a really dark region, and we'd see these sides as really light regions, because since this is thick, it doesn't let as much light pass through, hence it's dark. Alright, so now that we got the general anatomy going out of the way, and we know that muscles contract, follow me over to this side, and we're going to talk about some individual components. So I kind of broke down actin, which I know there's red, blue, and black, but this entire thing are in these little red lines right here. So there are a bunch of protein conglomerates inside this actin protein. You can use blue to label all of these. So they had the main core of an actin molecule is called nebulin. Don't need to know anything past that, just besides what it's called. And then these circles are actually the actin molecules, or like the actual actin molecules themselves. Despite we call this holding actin, the individual actin pieces are the circles. Now I did bother to differentiate between this, it's sort of like a helice, they're kind of winding around in each other. I did bother to differentiate the fact that there's two, because the individual strand is called F-actin, and that also applies to the red one, that's also F-actin. But when you combine them together, we call it G-actin. This black line that I have traversing just the red, it also traverses the blue, but I didn't want to draw it in because it would be too messy. This black line here is called tropomyosin. So if you think of nebulin as the sort of the core, the tropomyosin is like the exoskeleton. It's sort of just a structural protein that follows the outside of the actin. And bound to our tropomyosin and actin is this molecule called troponin. Troponin is made up of three individual parts, which we have diagrammed right here. So this is the troponin molecule on the actin molecule, which is part of the larger thing blown up to size. So the troponin molecule has three different parts to it. There's the I part, the T part, I should say binding site, I binding site, T binding site, and C binding site. The I binding site, this is where troponin binds to actin molecules. The T binding site is where troponin binds to tropomyosin. And then the C binding site, this is really the important part. The C binding site is where this troponin binds to calcium. So just as a note, the calcium is made up of 4 different regions. There's 2 low binding affinities and 2 high binding affinities of the C binding site. For the most part, the high binding affinities are actually taken up by calcium all the time. But the low binding affinity sites are going to be variable, meaning sometimes they're bound and sometimes they're not bound. When they are bound, our entire actin molecule is going to undergo a conformational shift where our myosin binding site, let me draw a larger actin molecule here. So let's say this is a inactivated actin molecule because there's no myosin binding site, but then calcium comes and binds to troponin. That binds there, and we're actually, this little circle on the back side is going to come, and this is where myosin, which is the blue part over here, is going to be able to bind to actin to be able to yank on it. When troponin has 2 calcium bound to it, myosin has super high affinity for binding to actin, and that works well. Talking about myosin, which again is the blue part over there, this is what myosin looks like in of itself. It kind of looks like a bunny. Not a really cute bunny, it's a bunny nonetheless. So the myosin has a number of regions, but this is called these trapezoidal things, is called the light chain region. And these are called the heads, the myosin heads. And when this is undergoing, or when it's actually binding to myosin or binding to actin right here, these heads are the part that bind. But this molecule in specific undergoes a conformational change as well. When it's getting ready to bind, it looks like this. ADP is bound and phosphorus is bound. And then we have our heads right here. And then when it binds, I drew that a little bit wrong. This is supposed to be straight up, like that. But then when it binds, it actually, and then contracts, it undergoes to a different conformation. And I'll detail this a bit later. But notice how we had a 90-degree angle here, and here we're going to have a 45-degree angle. And ADP and phosphorus will have fallen off at that point. Welcome back. So, we got some cells here. At this point in time in our propagation of action potential of our muscular fiber, what's already happened? We're not going to bother talking about it because we previously discussed it if you're a close fan. Our neurotransmitter has already been kicked out of our presynaptic neuron. The neurotransmitter is then going to go and bind to this ligand-gated channel. Now I can't specify that a neurotransmitter specifically is acetylcholine. That makes this specific channel something called a nicotinic 1 receptor, not to be confused with a nicotinic 2 receptor found between the preganglionic and postganglionic neuron. That's not what this is, but it is still a nicotinic receptor of the first class. So once the neurotransmitter binds to that, we are going to permit sodium to come in. And with enough stimulus, this will cause a depolarization of the sarcolemma. Once this depolarization occurs, we're going to get some Bwong action. So Bwong, 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 Bwong. and that is eventually going to depolarize, or rather activate, with this gate right here that is called a voltage-gated calcium channel. Now, here's where biologists failed at life, because they called it a voltage-gated calcium channel, but calcium doesn't flow through it. So, this is what it is. This voltage-gated calcium channel, we also call it the DHP channel. It stands for dihydropyridine. It's fun. But, point being is when this improperly named channel opens, it actually is attached to this mechanical channel called what's the channel called?
RYR.
Huh?
RYR.
Ryridine. RYR channel. We're going to go with RYR. This is called the RYR channel. Mechanically gated, so what that means is when this channel opens, it's actually really opening this calcium channel, or this voltage, or mechanical gated channel. That's not even called a calcium mechanical channel, it's just called the rear channel. And that's going to allow calcium to leave this bubble here called the sarcoplasmic reticulum. That calcium is going to leave, and it's going to go act on the actin molecules. So that way, we can do our whole process that we talked about previously, binding, contracting, super fun stuff. But while we depolarize, we might also run into an issue of we get such a high calcium concentration or potassium concentration, because remember when we depolarize, we bring in sodium and we also push out potassium. So this mechanism over here called the T-tubule, that's where this voltage gated channel is attached, and we also have ECF fluid in here, and we also have sodium fusing in and potassium fusing out. Point being is at some point in time we may get such a high concentration of potassium inside our T-tubule, and it's actually not wide enough to facilitate that high of a concentration. So it's sort of like the potassium channels to allow the leak potassium channels are saturated, meaning that the potassium, or there's just not enough of them, the same deal. The potassium is crowded, so it can't go back through, which means as we increase extracellular potassium, we're going to ultimately increase our resting membrane potential, which is a bad thing because we don't want to do that. That would cause our cell to be too excitable. So what does the body do about it? Well, guess what? The body thought about this and it planned for this. So we're going to take our chlorine ions, which we normally just ignore, and they are going to have leak channels in the T-tubules of muscles, but they're also going to have voltage-gated channels that allow chlorine to flow into the cell, which will therefore make the resting membrane potential more negative, returning it back to like it should be. I want to talk about a different concept now related to muscles, but how the contraction actually occurs. I kind of mentioned the components of contraction earlier, but what's actually happening between myosin and actin looks something like this. So this line is going to be our actin molecule. This is sort of our Z line, and then I'm going to draw my myosin like this. So this represents myosin. And in this part, we're actually going to start out with a phosphorylated, and therefore also has an ADP on it. So they're not connected yet, but we are going to undergo something called the cross bridge, or rather we're going to form the cross bridge for our myosin comes and binds to our molecule, to our actin. And then we're going to do the most important step, how contraction actually occurs, called the power stroke. So once the phosphorus falls off, although it's hard to show in a picture per se, phosphorus and ADP fall off, it's actually going to go back to a contracted state. So it was like this, and then it falls off and it goes like this. And in the process, you can imagine I had a rope right here. I pull the rope with me, and that's a contraction. It does that over and over again. To finish out that cycle, we're going to have ATP come in, and that's going to cause this dude to fall off. But lucky for us, Myosin has hydrolytic properties, which just means we're going to hydrolyze a phosphorus out of our ATP to form ADP. And when we do that, our myosin is going to go from the cocked position back to the open position, and it's free to contract again. And to top off this concept, we're going to talk about our sources of ATP throughout our body. So first and foremost, we have the cytosolic ATP, and that's just ATP floating around in the cytosol. Why is it there? Not our problem. But what we do know is it lasts about 5 seconds. Then we have a different source of where we actually create ATP. This is specifically inside our muscles. We have creatine phosphate. And essentially that means we have ADP and a phosphorus, and then we have a creatine kinase, and then that kinase is going to phosphorylate ADP and the phosphorus together to make ATP. That's going to last us about 10 seconds. And when I say 10 seconds, it means like if I needed to like flex my bicep, and as I'm doing a curl, if I go past 5 seconds, then it's going to convert to that, and I have 10 more seconds till I'm out of energy there. And then we go to glycolysis, which every Bio 180 student is so familiar with, and that's breaking down of glucose into pyruvates, and in the process of just glycolysis, we're going to produce 2 ATP. There's actually 4 produced, but we use 2 in the process, so it doesn't really matter. That's going to last us about 60 seconds of breaking down sugars that are nearby. These 3 I just mentioned are all anaerobic, don't require any oxygen. Then we go down to the big boy, oxidative phosphorylation. That's just that's going through the Krebs cycle and making NAD plus and FADH plus. And you're sending those down the electron transport chain, and that gives us a whole bunch of ATP, and that's going to last for hours. So like runners use a lot of this. Gym bros use a lot of this.